Hello, everyone, and welcome to today's webinar titled, How to Streamline Your Variant Classification Workflow with HGMD. This webinar is part of the Clinical Diagnostics and Research event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit them at QIAGEN.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature in the presentation. You can find that live chat at the right of your screen. You can also participate by submitting as many questions as you might have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just simply click on that Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the abstract window below the presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Malaina Gaddis, from the Clinical Field Application Scientist at QIAGEN Digital Insights. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Gaddis, welcome. You may now begin your presentation.
Hello everyone. I would like to welcome you to the QIAGEN Digital Insights webinar on how to streamline your variant classification workflow with the Human Gene Mutation Database, or HGMD. My name is Megha Balanadu, and I'll be your host and moderator for today. During the presentation, please use the Ask a Question box on the right of the slide window to post your questions to our speaker and QIAGEN experts, which will then be answered throughout the session. We will also have a Q&A at the end of the presentation. Please also check out our resources section for important links. With that, I would like to introduce our speaker for today, Dr. Malaina Gaddis. Malaina Gaddis is a clinical field application scientist at QIAGEN Digital Insights with previous experience as a variant interpretation scientist. And now I will hand the presentation over to Malaina. Thank you.
Thank you, Megha, and thank you all for joining today as we talk about different ways that HGMD can help streamline your variant classification workflow. I'd first like to go over the overview of what we're going to be discussing today. First, we're going to talk about what the Human Gene Mutation Database is, HGMD, and then also some advantages of HGMD Professional. And then I would like to walk you through some examples and use cases that I've encountered previously as a variant scientist that have really been good illustrators of how HGMD is a valuable tool to help with variant research. And then, as Megha mentioned, we will have a Q&A session. So first, what is the Human Gene Mutation Database? It is an expert-curated database of human germline mutations from peer-reviewed published journals. Drawing your attention to this figure on the right, you can see an analysis between the content in HGMD, OMIM, and ClinVar. And nearly 60% of the ClinVar entries at this point in time are found within HGMD. This remaining 36,000 is likely not in HGMD because ClinVar does not necessarily require users who are submitting their clinical assertions about a variant to have specific guidelines for evidence to support that assertion. And so we like to say if it's not published, it's not in HGMD. You can also see that HGMD has quite a bit of content that is not available in either OMIM or ClinVar. And so really, it's a great tool if you're asking the question, does this human germline mutation cause disease? I'd like to note that HGMD is for germline mutations only, not for somatic. Many of you are probably aware that there is a publicly available version of HGMD. I'd like to just take a moment and talk about some of the advantages of having a professional version. One of the main ones being that with the professional HGMD version, there are quarterly updates where information is recovered from more than 2,000 different journals. And this is done manually by genetics experts who are actually reviewing all of this literature. In this table on the right, I have some statistics between the second quarter release of this year for HGMD and the third quarter release. And you can see just between the two quarters, there's quite a bit of content, 20,000, 30,000 variants that are added just within one quarter. And I'd like to note that with the publicly available version, the content is 3 years out of date. And so you can see having this professional version, you get the most up-to-date clinical information for these variants. The HGMD entries will include the first example of the mutation where it was published in the effect. And additionally, there may be more reports, PubMed entries that are either functional studies or publications that indicate alternative phenotypes, information that may be useful to the user. And so there are a couple of ways that you can access this information. First off, with the advanced, with the HGMD professional version, you have advanced search capabilities that are not necessarily available with the public version. You have availability to search by gene or mutation. You can do phenotype searches. And you can look at these mutation overviews for specific genes to get an idea of the number of mutations by variant type. Also, there is an availability of a downloadable version of HGMD Professional. So if you prefer to have the data in tab delimited files or a MySQL relational database, that's also available. So now that we've talked about what HGMD is, I'd like to now move on to some examples where HGMD has been particularly useful as I've done variant research in the past. Specifically talking about, doing the research of a variant's clinical significance, some ways that visualizing variants through HGMD can provide information about the allele of interest that you're looking at, ways to identify genes associated with specific phenotypes, and then using some of the advanced capabilities to design an NGS panel. And so my first use case is for a variant in a gene that has kind of a difficult naming scheme. It's got legacy and alternative names that can make research kind of difficult. This gene is SERPIN-A1, and in this use case, a variant scientist would be given this variant to look over and to research. And given the HGVS, the Human Genome Variation Society nomenclature for this variant, I want to present what a workflow might look like. So for this variant, the user may go to ClinVar first. And because you don't necessarily want to take the entries in ClinVar at face value, you dig a little deeper into the entry and you see the associated literature for that variant. However, there's no mention of any alternative names or other schemes within this ClinVar entry. And so you're looking up this serine at codon 6 to change to leucine. And so you pull up this literature information that was associated with the ClinVar entry. And when you investigate this PubMed, you see that within this paper, the only serine to leucine change is happening at codon -19. You also notice that this is somehow also associated with something called PIZ rexum. So at this point, you're not seeing any mention of a serine 6 leucine. So you may move on to the gene review for SERPIN-A1, where you indeed confirm that alpha-1 antitrypsin deficiency is the associated phenotype. And you get a little bit of background information about the gene. You see that variants were initially described based on electrophoretic protein variants with the prefix PI denoting protease inhibitor. So essentially they would add this PI in conjunction with the name of the location where this variant was identified. And this was all before they knew that SERPIN-A1 was the associated gene. And so you have not only what appears to be like a legacy name where the codon is different, but you have this different alternative naming scheme based on the location of where the variant was found. But still at this point, you're not quite sure that you can confirm that serine 6 leucine is in fact the same variant as a serine -19 leucine. You take a look at the OMIM entry, and you kind of get the same information. The same literature is associated, the same names are given. However, you still don't have any confirmation that this is the variant you're looking at. You suspect it is, but to be certain, at this point, you would have to do additional digging to confirm that the information you're finding is relevant to the variant you're researching. I would propose going to HGMD as your first step in your variant classification workflow, because it's going to provide you that background information and save you time where you don't need to go and research these historic or alternate naming schemes. So here I'm showing a browser view of the online version where I've searched by gene, SERPIN-A1, and I've looked at missense variants. And within the first entry, I see the variant I'm interested in is serine 6 leucine. And conveniently, HGMD has a special column that describes the legacy change. And you can see that there's an offset by 24 amino acids explaining why you're seeing this -19. It was probably part of a translocation signal or something, and the original people studying started counting 24 codons downstream. And so I have confirmation, and I can click on this HGMD accession here to view the variant entry. And in this variant entry, again, I get confirmation that the associated disease or phenotype is alpha-1 antitrypsin deficiency. HGMD considers this a disease-causing mutation. Again, I have the codon number and the legacy codon number available in this view. And I have the primary literature report for the first time this variant was published, as well as an additional report. And so at this point, I can be confident that when I'm talking about this serine 6 leucine, it's the same thing mentioned in this primary literature, because the geneticists who are reviewing the information and updating the content within HGMD will not associate literature if there's any ambiguity. They will in fact follow up with the authors of the publication if there's any question. So they've done that background digging for you, saving you time so that you can move forward with your research. I have another example I'd like to show in another gene. This time you're looking at the gene NE, and you're looking at an arginine 297 glutamine change. And I'd like to break out of the presentation and show you a live example of how you could do this type of search within the online version of HGMD. Go ahead and log in, go to online, and you're first brought to the homepage for HGMD where you can see the current version, the contents, and any important announcements or updates. And so there's a number of ways I can search. I could search by gene, if I wanted to look at all the variants within the gene GNE and then narrow it down by missense, I could do that until I find the one I'm interested in. I'd like to show you another way where you can more directly pull up this information. Here in this mutation search, you can search by the codon. If you have the HGMD accession, you could directly look. dbSNP, I'm going to use the coordinate. So using the chromosome and coordinate, I can paste it in here. And important to remember that there's a drop down where you kind of specify how you're searching. And you also want to specify, if you're doing this by coordinate, which genome build, and I'm working in HG38. So I'll go ahead and I'll submit my query, and it directly pulls up my variant that I'm interested in. And so if I wanted to, I could also click on the gene symbol and get that same spectrum of mutations that I could find by searching this way. How I could link out to dbSNP. I'd like to review the accession. And so pulling up this accession, I see that the associated phenotype is sialuria. And again, the HGMD entry is citing this as a disease mutation. I don't see anything in the codon number about a legacy naming scheme. And so then my attention is then taken to the literature. There's a couple of different papers. The first one being reported in a Portuguese girl who had the clinical, biochemical, and molecular characteristics. Additional papers were added because the reviewers found that there was good information, useful information that they added. And I want to draw your attention here to the comments and the notes. Something that I have found particularly helpful at many times is that reviewers will add these notes that clarify things. For example, we're looking for something at codon 297, but within the paper, it's indicating that it's at arginine 266 glutamine. Again, another note in another publication, it's reported the same way, and it gives the ensemble transcript. Also helpful is many times the comment will tell you kind of where to look for the variant. So if it's only listed in the supplemental, they'll point you in that direction. And it also shows the value in having someone manually look at the information, as they're not just reviewing the text, the body of the text, they're able to look at, graphs and figures. They look at, supplemental table, supplemental information, and then they kind of point you to where they found that key piece of information for the variant. And so here, you know that there's a reporting scheme on a different transcript than the one you've been given. And so HGMD, the web version, has this additional predicted HGVS descriptor feature, where it will list all of the RefSeq transcripts, the HGVS descriptors, as well as the ensemble. And so you can match it up, that 815, and from here it gives the CDOT, but you can confirm that the 797 G to A is actually this 266Q if you translate it to amino acid. And so here's another example where, first reviewing the HGMD entry gives you key insights that will help you as you continue with your literature review. Now, if I were to go and look for additional literature, I would know that there's a possibility it could be reported a number of different ways. And so I need to be aware when I'm doing that search and how I form that search. And so, at this point, we've talked about how HGMD can help you with your variant research and save you time along the way. And so moving on, I would like to talk a little bit about some of the use cases for visualizing this data. and for downloading the data. So many people, when they download the HGMD VCF file, will it to the VCF from HGMD and annotate their variants. And one, use case for doing that is that, sometimes when you do sequencing, you'll get a large list of variants and you want to narrow down to look at those that are most likely to be clinically significant. And so you can filter by things like allele frequency or quality metrics, but you could also annotate with HGMD and filter down that way as well to come up with a smaller list of variants to look at in more detail. So another use case for that downloadable VCF is for visualization purposes. And so why is this helpful? Sometimes you want to see what's in the genomic neighborhood, what we like to call the genomic neighborhood. And this could be visualizing a VCF track for your variant of interest and being able to see if there are entries within HGMD that are at the same codon and that are marked as disease mutations or supposed disease mutations. And having this visual representation for many users is quite helpful. Other times you may find visualization helpful if you're looking at a particular region of a gene. For example, if you're at the 3' end and you're looking at frame shifts, and if you have something that's past the boundary for nonsense mediated decay, you need additional evidence from the literature or functional studies showing that that last part of the protein has significance if it's cut off. And so being able to see visually an HGMD track showing other reported disease mutations in that location will point you to some of that evidence. Another use case that I find helpful is having the HGMD track loaded to help confirm that my secondary analysis is accurately calling or describing variants. So I have a case here that I've uploaded into the UCSC genome browser. It's within the APC gene, and I'm at a region where there's a little bit of repetition with this AG motif. And my variant of interest is here in purple. And you can see that there's a little dash on top followed by the GA on the bottom, indicating that you have an assertion of a GA. And I can see by loading the HGMD track on top, which is what all these variants on the top are, in blue, I have an insertion of a GA that's nearby or in the genomic neighborhood. And so as a person, reviewing this variant and maybe examining the bed file or the BAM, the reads, I might suspect that this insertion is in fact supposed to be called here, a few bases away, downstream. Because in repetitive regions, sometimes there can be changes in how it's represented in the VCF versus HGVS. And so I would know to follow up maybe with Sanger confirmation to make sure that this is in fact the variant I want to look at and that it doesn't need to be altered in my secondary analysis to actually reflect something that's been reported in the literature as a disease mutation. So you can see how having this visualization makes this type of analysis quick. And just briefly, I'd like to go over kind of how you'd go about that process of uploading. So when you have a professional license for an HGMD download, you have a number of different files that are available to you. I'm highlighting here the VCF, the zipped VCF in HG38, also available in HG19. So you would just download that. And then many individuals will use Integrative Genomics Viewer, IGV, or the UCSC Genome Browser. A lot of times personal preference. Just to give you an idea of what an upload of this VCF looks like in IGV, I've loaded it here, and then, I've blown up the information here. So this is information parsed from the VCF file. And so you can see the gene and the phenotype, the HGVS nomenclature, maybe the VCF format is not supported as well in IGV. It has this little percentage, but you can get the key information here. I tend to prefer using the UCSC Genome Browser. I like that you can see the nature of the variant change. So you can see if it's a deletion or if you have an insertion. You can also see if you have, like G to T, large CNVs. It provides just a little bit more information. And so this is kind of what the parsed information from the VCF would look like. We do have versions that are specifically formatted for the UCSC Genome Browser that provides the full entry. That's available with our Genome Tracks product, just as an FYI. And so I have some additional use cases that I'd like to actually show. So I'm going to move out of PowerPoint. And my first one, I'll pull up. So just briefly, it's relatively easy to upload a VCF within the UCSC Genome Browser. You would go to My Data, Custom Tracks, and then within here you would add custom tracks, and then there's just a quick upload. And this, you can save your session, so if you don't want to have to reload every time you pull up the information, it will save all of these settings and configurations. So I have here in this APC variant that I initially showed, here is my variant of interest in purple. There's a dash at the top, so an insertion of an AG in this repetitive region. And so I can load my reads file and determine if my variant calling pipeline needs to be adjusted or if this is in fact a novel variant in the individual. Another use case for having this information in this visual format is if you're doing like variant classification according to ACMG guidelines. One piece of information or evidence, here I have my variant of interest in the gene PAH, which is a G to T. I can quickly see that at this same location, there have been disease mutations reported in HGMD for G to C and G to A. And so this is something that as part of ACMG, you can use as a piece of evidence if these are then shown to be pathogenic. That tells you that, this codon has some sort of functional importance. And so having this kind of neighborhood view to see kind of what's upstream or downstream can be very helpful at times. And so, now that we've talked about some of the applications for using a downloadable VCF, I'd like to move on to some other use cases. Here, for when you may want to identify genes associated with a specific phenotype. If you're more on the research side and you're interested in discovering what genes are associated with a phenotype, in this case, bipolar disorder, within the online user interface, we have the ability to search by phenotype. So then all I would have to do is I could do a Boolean search where it will pull in the actual entries with the term bipolar disorder and search within HGMD phenotypes. And I can quickly find, for the associated bipolar disorder phenotypes within HGMD, a list of genes that have, associated literature indicating some sort of relevance or impact with this phenotype. And so this is a good place to start if there's, additional places I want to take my research. I have this list to follow up on. Another use case might be to utilize the content within HGMD to design a panel, maybe based on phenotype. Maybe I want to design a panel for cardiomyopathy. So in this case, you would do an advanced search. And I'll actually do kind of a demonstration showing the search, but what you would get is a list of the variants within HGMD that are associated with cardiomyopathy. Oops, sorry about that. And so I'll skip out now and show you kind of how I did that search. So I went to advanced, and within advanced, I went to nucleotide substitution plus. And within this page, I'm interested in searching by disease phenotype, but you have a number of different parameters that you can include in your search. If you wanted to search by functional elements, maybe if you're doing research and you want to look at variants within known transcription factor binding sites, that could be particularly useful. Motif search, if you're looking for specific, splice variants or regulatory regions. I kind of want to look at it all. So I've highlighted my output file, the information that I want to receive back. I want to get back mutation, variant, dbSNP, and I've specified my genome build. And so I'm just going to specify cardiomyopathy and then hit submit. And at this point, it pulls up that table, and I can actually download a bed file that you can then load into UCSC, and then you can visualize across all of the associated genes with cardiomyopathy, the known variants. So if you then wanted to go and design targets for a panel, you can make sure they're covering all of these associated HGMD entries. You can also click here to save it as a text file. And so then you pull this up, probably look better in Excel, but you have all of this information that was brought forward, which is displayed here in nice tabular format, but you have that option to export it and save it. And so hopefully, I've been able to show that HGMD has a lot of utility for variant classification, and that it can really speed up your process by giving you that information about historical or legacy names, different transcripts, so you can quickly get through your literature search and get to your answer. And, there's different ways that you can use the data to visualize or to annotate, to help with other aspects of analysis. And you have many different options for searching for this information, by phenotype, and that advanced search where you can actually export and then move on to help design a panel based on a phenotype. So, thank you for your attention and for listening, and I'd be happy to take any questions at this point.
Thank you for that informative presentation, Malaina. With that, let's straight away jump into Q&A. I see a lot of questions coming. My first question would be, how often is HGMD updated?
So HGMD is updated on a quarterly basis.
Okay. Second question, is there an uploadable VCF with more information included from the HGMD entry?
Yes. So with the download of genome tracks, which you can license, you have a number of different files that are specifically formatted for upload into UCSC. And there's different ways you can view the HGMD. You can view by like phenotype and by disease mutation or functional polymorphism. So yes, that is definitely available.
Great. Next question would be, does HGMD provide information about genomic polymorphisms in genes?
Yes. So for a polymorphism to be included in HGMD, there is some sort of literature evidence showing a link to disease or a phenotype. And so polymorphisms can be added to HGMD.
Okay. We have some time left to take a few more questions. Next one, which genome build is used for HGMD?
So HGMD has been updated for HD38, and most of the information is presented that way. However, there is still support for HG19 so that you can also see that information if that's the build you're using.
Okay. Why are there articles for a specific variant that have not been captured in HGMD but can be found using other search tools such as Google Scholar, PubMed, etc.?
Right. So you will note that there are just a few articles shown, and really the aim of HGMD is to show the primary report, that first report where the variant is published in the literature. And the additional articles are really only added if there's something new or important that they add. For example, an updated phenotype or functional evidence talking about, protein structure. And so the goal of HGMD is to not necessarily return all of the possible information available for a variant, but to give you that key background that's important. And so that's why you're going to find those key pieces of information rather than every single publication for that variant.
Next one. Do we have to pay extra to download the data from HGMD if you already have HGMD Pro?
Yes, so the HGMD online version and the HGMD download are both part of different licenses, and so you can consult with a customer representative to see what the price difference may be to add the download to the online version of the license.
Okay, we have 3 more questions. In HGMD, does it provide the information or evidence of its variant classification?
So HGMD is technically not classifying variants in the terms of ACMG, but they will give an annotation of a disease-causing mutation or a DM, or if it's suspected to be a disease-causing mutation, it'll have DM ? And so the evidence is within those publications. And the curators who are reviewing this evidence always err on the side of adding something rather than leaving it out if there's any question about its association with disease, so that if future publications come out, this variant is kind of flagged for further review so that it can be, revised in the future as more evidence becomes available.
Great. Should we be refreshing VCF's upload from HGMD to UCSC every quarter?
Yes, so every quarter when the new version of HGMD Professional comes out, there will be substantial additions and new mutations that are added to the database. So I think it would be a good idea to upload the new VCF each quarter, just so that you're reflecting those new additions to HGMD.
And the last question of the session, is HGMD integrated into QCI? If so, is it the full version of HGMD or the public version?
Yes, so QCI is QIAGEN Clinical Insights Interpret, and it's a software that assists in variant interpretation. And so with the license of QCI Interpret, you are able to see the full HGMD entry for the variant that you're looking at. So what this means is if you wanted to search for nearby variants, those HGMD entries wouldn't necessarily show up as part of QCI Interpret. You would need the additional license. But for every variant in your VCF that goes through Interpret, for which there's an HGMD entry, you will be able to view that entry as part of Interpret.
All right. Before we conclude, I would like to thank our speaker, Dr. Malaina Gaddis, and most of all you, our audience, for your attention. We hope this was informative and helpful. However, if you should have any remaining questions, please do not hesitate to contact us. We are here to help you. This concludes today's session on HGMD. We look forward to hosting you in future QIAGEN Digital Insights webinars. Have a pleasant rest of your day. Thank you.